So today, we will learn about the bilirubin formation and metabolism. So, firstly, let's talk about what is bilirubin. Bilirubin is a yellowish compound that is produced when body breaks down erythroid cells or hemoglobin, which accounts for 70 to 80% of bilirubin formation and other heme-containing proteins such as myoglobin, and cytochrome, which accounts for 20% of bilirubin production. Now let's see how bilirubin is formed from heme proteins. As we know that red blood cells flowing through the blood vessel have an average lifespan of 120 days. Inside each red blood cell, we have heme molecule and a globin chain. The globin chain gets recycled to amino acids. Now let's see the heme molecule. Each heme molecule is made up of a ferrous molecule and 4 pyrrole rings. The ferrous molecule gets transported in blood as transferrin, and pyrrole ring act as a substrate for formation of bilirubin. So as the fragile red blood pass through the reticuloendothelial system, their cell membrane ruptures and released hemoglobin is phagocytosed by macrophages. As already told, the pyrrole rings are first converted to biliverdin by enzyme hemoxygenase and finally to bilirubin by enzyme biliverdin reductase. This free bilirubin, which is gradually released from macrophages into the plasma, immediately combines strongly with plasma albumin and is transported in this combination throughout the blood and interstitial fluids. So, even when this free bilirubin is bound to albumin, this bilirubin is still called free bilirubin. This free bilirubin is highly toxic. Now, within hours, the free bilirubin is absorbed through the hepatic cell membrane. Hepatic processing of bilirubin occurs in endoplasmic reticulum and involves conjugation of free bilirubin with 1 or 2 molecules of glucouronic acid by enzyme UDP-glucuronyltransferase and formation of bilirubin glucuronide, which is a water-soluble and non-toxic form of bilirubin. Now the conjugated bilirubin is transferred via bile to small intestine. As it enters large intestine, it gets converted to colorless compound called urobilinogen, and this urobilinogen gets self-oxidized and converts into yellow-colored compound called stercobilinogen, which gives feces its characteristic color. Now the conjugated bilirubin is transferred via bile to small intestine. As it enters large intestine, it gets converted to colorless compound called urobilinogen, and this urobilinogen gets self-oxidized and converts into yellow-colored compound called stercobilinogen, which gives feces its characteristic color.